All right, good afternoon, everyone. We're going to talk about some chapter 23, just really population evolution in general, right? So let's go ahead and move on. Now, your chapter kind of opens up with this finch, right? And the point is, just looking at that finch, can you tell whether it is evolving or not? Basically, no, right? So you can't really tell whether a particular individual within a population is evolving. It's an entire population that can be observed as evolving, and it takes a long time, right? So that's the point here. It's individuals do not evolve, populations do. Now, let's talk about microevolution. So remember in the beginning of the semester, we talked about macroevolution. Well, microevolution is changes in allele frequencies. Remember alleles. So alleles are a particular form of a gene. And in diploid organisms, they have two of each alleles, or two alleles. And the allele frequencies are going to change in populations over through each generation. Now, there are three main mechanisms that cause these changes in alleles frequencies. It includes natural selection. We talked about natural selection. I'm going to kind of review and give some examples of this. Genetic drift and gene flow. Now, natural selection is the only type of mechanism that is consistent when it causes adaptations, right? So adaptive evolution. Now, genetic variation is basically the basis of basically the start of evolution, right? So there must be genetic variation present for evolution to actually occur. Because remember, in evolution by natural selection, in terms of natural selection, something needs to be selected for. Now, if everyone had the same traits, then there would be nothing to select for or against. Now, variation in heritable traits is a prerequisite for evolution. So for evolution by natural selection to occur, it must be a trait that is heritable. Now, at the DNA level is where most genetic variation comes from. Now, genetic variation at the DNA level can happen in the genes, or it can happen in other DNA segments that are not genes. Okay, so remember the genes are code for protein, and then there's segments of DNA that do not code for protein. Now, there's difference in terms of genotype and phenotype. So there's a little review of General Biology 1. Okay, so remember phenotype is the product of an inherited genotype. It's also partly influenced by the environment. Now, it's the inherited part that natural selection can generally act upon. Now, natural selection can only act upon variation that is heritable, and I've kind of already mentioned that. Now, natural selection acts directly on the phenotype, but indirectly on the genotype. Even though it's the genotype where the alleles are, it's the phenotype that's acted upon. Think about this. If you were choosing a mate, do you look at their DNA to choose a mate? Probably not, right? So most animals and other organisms, they're going to choose their mate based on some sort of phenotypic traits that they prefer, right? So what if it's a tall individual? What if it is a nice individual, right? So personality kind of matters sometimes too. So again, you're going to select based on the phenotype directly, but indirectly that affects the genotype. Now, genetic variation can be measured in a couple different ways. It can be measured as gene variability or nucleotide variability. They kind of are dependent on one another. Okay, so gene variability is measured as what's called average heterozygosity, or the percent of heterozygotes in a population. So increased heterozygosity, right? So if you see an increase in the percent heterozygotes, then this can equate to an increased variability within that population. Okay, so they're kind of, they work together. Nucleotide variability is directly is the DNA sequences, so changes in the DNA sequences. Most variations occur in the non-coding regions. So remember, I told you, not in the genes. So it happens in the genes, or it can not happen in the regions that are not gene material. These are neutral. It can also be neutral if it is a gene or in the coding region if it doesn't change an amino acid. So remember, if it changes an amino acid, this affects the structure and the function of a protein. That ultimately affects the fitness of the organism. Okay, so those that are neutral are ones that are going to occur in the non-coding regions or going to result in an amino acid that is the same as the normal. Now, let's talk about some sources of genetic variation. Right, so there's several sources of genetic variation. How does this actually occur? So new alleles can be formed by mutation. Okay, so mutation really is the ultimate source of evolution. Alterations in gene number or the position of the gene. Okay, the position of the gene can be caused, and the gene number can be caused by duplication events. Or chromosomal aberrations like transversions or inversions. Deletions, substitutions, insertions can all affect the genes and the alleles. So there can be an increase in the mutation rate or decrease in the mutation rate. Generally, an increase in mutation rate is going to occur with rapid reproduction. So those organisms that can reproduce very quickly are generally going to have a higher mutation rate. Sexual reproduction can introduce like combinations of alleles that already are present, right? So remember, crossing over, that occurs during prophase 1 of meiosis, right? So that's recombining alleles that already exist. Now, formation of new alleles can arise by mutation in terms of the DNA sequence. So here's an example, okay? And there's a difference between mutations in the germline and mutations in just somatic cells. So the germline mutations are going to be, let's say, in the sperm or egg. So here we've got an example of a germline mutation. If you look on the left here, okay, so we got a germline mutation, all right, in the sperm. Okay, so this is going to be carried out, and then basically this mutation is going to exist in the entire organism. Okay, so the whole organism is going to have the mutation. Now, if there's a mutation in the somatic cells, this could be caused by just a random mutation during duplication of the cells or repair, or also by chemical changes, right? UV irradiation or other types of chemicals. Okay, it's only going to affect the area where the mutation occurred. Okay, now this could happen as an embryo or it could happen as an adult. Okay, so a germline mutation can have more an effect than a somatic cell mutation. Now again, it depends on where the mutation occurred and at what time. All right. So the effects of point mutations can vary, and I already kind of talked about in terms of development. Now, mutations can alter the phenotype. Okay, this is usually harmful. This is usually not a benefit. Okay, harmful mutations can be hidden from selection in recessive alleles. Okay, so the heterozygotes. Okay, they can be hidden in heterozygotes. Mutations that result in a change in the phenotype are often beneficial. If they're beneficial, it's usually rare. Okay, so if it is beneficial, it is extremely rare. Now, point mutations in non-coding regions are generally neutral, and I already kind of talked about that. This provides no selective advantage or disadvantage. It has no effect on the fitness of the organism. Okay, so the term for that is neutral. Now, remember, those mutations that can occur in the genes, if they're neutral, you would expect no change in amino acid. Now this can easily happen, right? So even in genes, remember the triplet codons, right? So if we look at the codon table, we've got, let's see, 1, 2, 3, 4, 5, 6 codons which code for the same amino acid. So there can be changes in bases, but still no change in the amino acid. Okay, so that would be considered neutral. Now again, if the amino acid changes, it's usually not neutral, and it's usually not beneficial. All right, so altering the gene number and position, remember these are at the chromosomal level. Okay, so you can have mutations that delete regions of a chromosome. They can be rearranged, right? Or just disrupted. Let's say, well, disrupted by, it could be a deletion, or it could be some sort of inversion. There could be a section moved from one chromosome to another. Again, these are typically harmful. Duplication of small pieces of DNA generally increases the genome size, of course, right? So if you're duplicating DNA and it remains in the genome, of course the genome size is going to increase. This is generally less harmful. Now, duplicated genes can take on new functions by further mutation over time. So once a gene gets duplicated, one of them can keep the same function, but then the other one could change. Of course, they're both going to change over time. They're going to both start to diverge. They're going to change over time. And one of them can take on a new function. So an example would be odor detecting gene. Let's say there was only one in the ancestors, in our ancestors and mice ancestors. Okay, it's duplicated many, many times. Humans have 380 copies of the gene, and mice have 1,200 copies of the gene. Could this allow mice to basically detect new types of odors? Right? So let's say it started out with one gene, right? And now you have many copies, and each of those copies are going to diverge and change, taking on new functions, like detecting new types of odors. Now, in terms of reproduction, mutation rates are generally low in animals and plants because the generation time is quite long. Okay, there's generally only one mutation in every 100,000 genes per generation. Mutation rates are often lower in prokaryotes. However, short generation times allow the mutations to accumulate rapidly. Okay, so this increases the mutation rate. Okay, sexual reproduction or asexual reproduction results in the ratcheting up of alleles that can be deleterious. This is called Muller's ratchet. Now, viruses both have a high mutation rate and short generation times. So their evolution is relatively uncontrollable. And we're kind of seeing those effects today. All right. So sexual reproduction can introduce new alleles and new combinations of alleles. So those organisms that can reproduce sexually, this is really where most of the variation comes from. And it comes from the recombination of alleles. Now there's three sources of variation in sexually reproducing organisms. Okay, remember crossing over, I already kind of mentioned that. Now remember independent assortment. Okay, so how the chromosomes line up at the equator during cell division. This can allow more variation. And then random fertilization. So remember there's billions of eggs, billions of sperm. They're all different. Each of them have different combinations of alleles. Which ones come together, right? So we have an egg and many sperm, okay? Which egg happens to be present at that time? Which sperm happens to reach that egg and get in there and fuse with it, okay? Completely random. All right, so natural selection, genetic drift, gene flow can alter allele frequencies. Now, These are the three factors that cause allele frequency changes. Okay, so natural selection, genetic drift, and gene flow. Now, genetic drift is basically describes how allele frequencies just fluctuate randomly. It's not predictable from one generation to the next. Okay, so this happens in all types of populations, although smaller populations have the greatest effect of basically genetic drift affects some small populations a lot greater. Now, it tends to reduce variation. Okay, so variation tends to be reduced when genetic drift takes place. Often alleles are lost. And again, I already mentioned this, small populations are affected more drastically by genetic drift. The smaller population, the greater the chance of deviation. Now, if we look at an example of genetic drift in this small population of flowers, right? So we have three phenotypes. We have the pink, we have the white, and the red. The pink ones are the result of incomplete dominance. Okay, so those are the heterozygotes. And then we have the white and the red. Those are the homozygous conditions. So we start out at a frequency of, again, P is the frequency typically of the dominant allele. Q is typically of the recessive allele. Okay, so it represents the frequency, and the frequency is the total, the number of individuals, okay, with whatever phenotype, so let's say number pink, divided by the total population. Okay, now number of alleles, or number of present alleles, okay, the pink alleles, okay, and this could be the genotype or just the allele frequency. So again, frequency often is shown as this right here, and it could be whatever. So in this case, this would be a frequency of the heterozygote. So I'm going to go ahead and just put RW there, make it a little bit easier. Now, the highlighted ones are the ones that are going to be contributed to the next generation. Okay, so only, well, we've got 3 pink and 1 white and 1 red. Okay, so this is the result. So after this, right, so only those are contributed. If everyone contributed, the population frequencies would probably be the same. Okay, but only certain ones did contribute to the next generation. This happens in most populations. Not everybody reproduces, right? So now we have frequencies of 0.5 and 0.5 for the dominant and recessive alleles. Okay, so let's say only these 2 red ones, reproduce in the third generation. Now we're only going to have red individuals. So what happens is the frequency of the dominant allele is 1, and then of course there's none of the other one, and we've lost that allele. So when there is a frequency of a dominant allele of being 1, so if everyone has it, it's said to be fixed. Okay, so that allele is fixed in this population. All right, so the founder effect and the bottleneck effect are kind of closely related to genetic drift, okay, because we're dealing with small populations here. Now, the founder effect occurs when a few individuals become isolated from a larger population. So they still are exist in the larger population, but they're not mating with anybody randomly in the larger population as a whole. Okay, so what happens here is the allele frequencies in the smaller population, which is called the founder population, will change and be different than the original population. So if we look at this right here, we've got this whole population, okay, and then we've got a subpopulation which has isolated themselves from everyone else. Okay, so they're just mating with each other now. This is the founder population, or founding population. Over time, all the descendants of the founding population are only going to have these 2 alleles, where the rest of the population has mixed alleles because they're mating with each other, right? So they haven't isolated each other or themselves. Now, the bottleneck effect occurs when there's a sudden reduction in the population size, usually due to a change in environment. So an example might be, let's say, a meteor strikes the Earth, right, and decimates most of the population, and only some people survive. Okay, so that surviving population is the population that's going to be contributing to the next generation. Okay, so here the gene pool is going to be reduced significantly. Okay, so again, this is very similar to the founder effect. If the population remains small, it will be affected by genetic drift. Remember, genetic drift has its greatest effect on small populations. And here is where a lot of alleles can be lost and alleles can be fixed. Right, so let's look at an example of the bottleneck effect with an actual bottle, right? So a bottleneck is ultimately a constriction somewhere, right? So here we're talking about a constriction in population size. Okay, so let's say we have an original population. We've got the blue, yellow, and white alleles in here. So let's say now we have some event that is basically going to reduce the population size by most of the bottle, because most of the individuals are not going to make it past this bottleneck right here, whatever that might be, right? So then basically the surviving population only have these alleles here. So they're going to mate with each other. And as long as that population remains small, some of these alleles may become fixed. Some of them may be lost. Okay. Now this population can get much larger and larger, and over time they can diverge. Okay. So the allele frequencies and new alleles may arise. So let's look at the effects of genetic drift as a whole before we move on to the rest of the stuff. All right, so genetic drift is again, significant in small populations. It's very important. So remember, the founder effect and the bottleneck effect result in small populations becoming isolated. Genetic drift can cause allele frequency changes at random. Okay, so these are just random changes in allele frequencies due to population size. Genetic drift can also lead to loss of genetic variation within the population. So with the loss of genetic variation goes along with alleles becoming fixed. Genetic drift can cause harmful alleles become fixed. Okay, so often harmful alleles are weeded out of the population if the population is large enough and there's enough mixing of alleles. Now, if this doesn't happen because it's a small population, then of course, the harmful alleles can become fixed. All right, so let's talk about gene flow. Gene flow is just basically the movement of alleles among populations, and this could be the movement between populations and within a population. Now, alleles can be transferred through the movement of actual individuals or gametes. So an example would be pollen. Okay, water could help allow sperm from like plants to move around, right? Or just individuals, right? So we have planes, trains, and automobiles, right? So as a whole organism, rather than just the gametes, we can move from one continent to another as humans and be able to, transfer alleles in that way. Now, gene flow tends to reduce variation, okay, among populations over time. Now, initially, it can introduce new alleles and basically increase variation. However, over time, it tends to reduce variation. Now, gene flow can affect adaptation to local environments. Because we have the mixing alleles, and whichever one's mixtures kind of benefit for that particular environment are going to be more likely to survive and reproduce. Now, let's look at an example. Okay, so we have this variation in a banding pattern of these water snakes. Okay, so we have those on land, and then those adapted to the islands that are around the area in the Lake Erie area. Now, on the mainland, this is where most of the banding is. So they're very strongly banded, and then on the islands, they're either unbanded or somewhat intermediate. The unbanded snakes are better camouflaged in the island habitats. So the lack of banding or reduced banding is selected for in the island and not selected for in the mainland. So if we look at some data, Okay, so again, mainland are banded, the island environment, they are unbanded. Okay, so some data show a movement from the mainland to the island, right? So started out banding, and then over time, right? So they mated on the island, and then the unbanded seem to be preferred. Now let's look at this. A through D here. Okay, so we have A, B, C, D, A, B, C, D, A, B, C, D. Okay, so A represents the most banded, D represents the least banded or unbanded. Now here we have data from the Ohio mainland. So we have Ohio here. Okay, so that's part of the United States. And then we have the islands, which are somewhere in between, right? In between the United States and Canada, of course. So we have the Ontario mainland, which is Canada. Now look at the frequencies here. So this represents the percentage of individuals, right, out of 100%, which are either highly banded or all the way reduced to unbanding. Okay, on the islands, you can see that the lower banding amounts have increased in frequency. Right, in Ontario, the banding is selected for, which is quite interesting. Right, so continued migration of the banded snakes, right, from either the mainlands to the islands would basically change the allele frequencies on the island. So again, this continued migration of these are going to maintain the alleles for the banded pattern even in the islands. So here we could see some are still banded, but it's because of continued gene flow, right? So gene flow tends to reduce variation. What do you think's happening in Canada? There must be not much gene flow in the island to Canada. There must be gene flow between the United States and the islands more so than any flow between Canada and the islands. All right, so gene flow can increase the fitness of a population, right? Increase the fitness of a population. Consider this example. So the spread of alleles that provide resistance, right, to, for example, insecticides. So insecticides are often used to target pest insects like mosquitoes, especially those that carry any viruses or even malaria. Now, alleles have evolved in some of these populations that allow the insects to survive in the presence of the insecticide. So basically, the insecticide has little effect on them. So the flow of this insecticide resistance alleles, right, into a population can increase that population's fitness because it's a benefit to have the allele that provides resistance to that insecticide. So that's also going to increase the frequency of those alleles in that population as well. Now, natural selection. Remember, you should already know the definition of natural selection. Okay, so this, in terms of alleles, it results in alleles that are being passed to the next generation in proportions that differ from the previous generation. An increased proportion or a decreased proportion, right? So if it's selected for, if it provides a benefit, then it'll be an increased proportion. If it doesn't provide a benefit, it will be decreased in the population. Now here, if we kind of go back to resistance to DDT in fruit flies, okay, so DDT is another type of insecticide. Now, an allele that confers resistance to DDT in the fruit flies, this is going to increase the proportion of the frequency, basically increase the frequency of that allele because it provides a benefit. Right, it allows these flies to survive in the presence of DDT. So again, as they reproduce, they're passing on this allele to the next generation because the ones with that resistance gene or allele are more likely going to survive and reproduce in the presence of that selective pressure, the DDT. All right, so natural selection can cause adaptive evolution. Adaptive evolution really is a process in which traits that enhance the survival or increase the fitness of an organism, right, increase over time. All right. So, natural selection, in terms of adaptive evolution, it has provided some interesting adaptations over the course of time. So, look, certain octopuses can change color rapidly, right, to camouflage them. Look, you could see his eyes right here, right? So he can camouflage very, very well at the bottom of the sea. Another example would be the bones of these within the snake, the head of the snake. So they've evolved to become like movable so that it can allow the snake to open its mouth to swallow prey that's much larger than its head. Right, so natural selection is the only mechanism that consistently causes adaptive evolution. Evolution by natural selection involves chance, right? So there's some a mutation that occurs, right? But then it's sorted out. Okay, so then it's sorted out. Who are the winners and who are the losers, right? So new genetic variations can arise by chance, right? So then those that have this mutation, let's say the mutation is a benefit in their particular environment, it's going to be selected for by natural selection. Okay, so natural selection consistently increases the frequencies of alleles that provide an advantage. Now, this is basically a summary, right? So I kind of introduced, the three things that affect allele frequencies the majority of the time. So I kind of introduced different forms of variation. We did a little review of mutations and different types of mutations and where this variation can come from. A little review of genetic drift and gene flow, and of course, natural selection. All right, thank you for listening.